I've got 2 o'clock, so we will go ahead and get this kicked off. I'm Chris Butts with Forest Products Society, and I would like to welcome you into our monthly webinar series, Forest Products Insights. Each episode of Forest Products Insights will welcome a guest speaker sharing their research, technical expertise, and experience to a live audience. The webinar is meant to be an open conversation where your questions can be answered and ideas can be shared amongst peers within the forest products sector. Please feel free to enter your questions into the chat box at any time, and we'll have time at the conclusion of the presentation for oral questions as well. If you're not an FPS member, we also invite you to learn more about Forest Products Society and consider membership in our organization. Today's speaker is Dr. Adriana Costa, a botanist with over 20 years experience in wood identification. She is a 4-time graduate of the University of Sao Paulo, where she holds a Bachelor of Science in Biology, a Bachelor of Education in Biology, a Master of Science in Wood Science, and a PhD in Botany. As a professor in Brazil, she taught university courses in plant anatomy and morphology for 6 years. And at the USDA Forest Products Laboratory in Madison, Wisconsin, she spent an additional 5 years conducting cutting-edge research in the area of forensic wood identification using methods such as DNA, computer vision, and near-infrared spectrometry. She is currently pursuing her 2nd PhD in sustainable bioproducts at Mississippi State University. Her topic today is organellar microcapture to extract DNA from recalcitrant wood specimens and trace evidence. We hope you enjoy the presentation, and we'll now turn things over to Adriana. Thank you.
Thank you, everyone. I'm going to share my presentation now. Can everybody see it?
We see it. It's not in presentation mode yet.
About now?
Perfect.
Is it still in presentation mode?
Okay. It is. Yep.
So thank you so much for having me here at the Forest Products Insights. I'm really excited to be here today sharing more about this work on DNA extraction from wood. So Chris already introduced me, and I just want to add that I'm really excited to be here at Mississippi State University doing a 2nd PhD in sustainable bioproducts. It is a great opportunity to integrate my botanical background and develop a more practical and applied set of skills to solve problems for the wood industry and for academia as well. So this is what I'm going through today. I want to show you the problem that we want to address and the obstacles that resulted in our question. Then I'm going to talk about the methodology that we developed to overcome these obstacles, and I'm going to show you the results at the same time as I'm talking about the technique. And lastly, I'll mention some applications of this technique. So what is the problem we want to solve? Our problem is how to identify wood. So we have a need to identify species, provenance, and the individual tree in forensic wood anatomy to combat illegal logging and to ensure sustainability. So the only potential tool that has a promise to solve all of these 3 needs is to use DNA-based technologies. And why DNA? Well, one of the reasons is because we can just pluck leaves out of a tree and use it to calibrate this method without having to kill trees. And we already have a global database that is being fed with new sequences every day. For example, other methods such as chemical, like DART and near-infrared spectroscopy, they not only need wood to calibrate the method, but they also need hardwood to build a database. And these databases are just at the beginning. So this was our problem. We have a need to identify wood in different instances, and DNA is a promising approach. But what are the obstacles? So although we can use leaves to build a database, and we already have a huge database, we don't know how to get good amount and quality DNA out of wood. So but when I say that we don't know how, it means that we have to adapt methods developed for leaves, and because there is no such thing as a standard protocol to extract DNA from wood that works for everything and every time. And I will show you why. As I just said, we don't know how to get good amount and quality DNA out of wood. So these are, for now, these are our two obstacles. And just to illustrate, let's compare the amount of DNA in leaves and wood. Here in these two illustrations, we have a leaf cross-section in the left and a wood section on the right. In the leaf illustration, we can see all of these green cells. Here are the upper and the lower surface of the leaf, and all these cells inside are ones involved in the photosynthesis. These brown structures are the veins in the leaf, and they are responsible for water and nutrient transport. And everything that is green with a blue circle is a living cell. The blue circle is the nucleus, and that is where the most of the DNA is in the cells. Some of you may remember that plants also have mitochondria and plastids, and these organelles also have DNA. So plants have basically 3 different genomes. But in this presentation, I'm only going to talk about the nuclear DNA, although we can use this technique for plastids as well. So in the veins, there is the tissue that transports water. And these are empty tubes with no cell content, and they have no nucleus. So moving on to the wood, and because wood is a water conducting tissue, the vast bulk of the cells are also tubes with no cellular content and no nucleus. The only living cells in wood are the parenchyma cells. These are alive and have a nucleus containing DNA. So I'm going to mention this word parenchyma again in some of the following slides. So keep in mind that these are living cells with no nucleus. And then considering that the blue circles are nuclei, it's evident that leaves have more DNA than wood. So we just saw that the major tissue in wood is a vascular tissue formed by dead cells, and these cells have no cellular content and therefore no DNA. Another problem that when considering wood as a source of DNA is that there are extractives in wood that interfere with the DNA extraction and inhibit one of the steps that we call PCR. I'll explain it soon, where we copy a portion of the DNA. And lastly, when we consider commercial wood or wood products, they are usually exposed to industrial treatments or processes that damage DNA. For example, the high temperatures used in kiln drying processes. So now we have 3 obstacles. In summary, wood is not the best source of DNA because of reduced quantity, recalcitrance of wood, and reduced quality sometimes. So how is the reduced quantity of DNA affected by the current DNA extraction methods? To extract DNA, we usually rely on DNA extraction kits. There are some kits that are generalist and can be used for any kinds of cells like animal, plants, and fungi. And there are other kits that are optimized for plants, but they are developed specifically for leaves. These kits require around 50 to 100 milligrams of starting material, depending if the tissue is dry or fresh, and it's around 5 to 7 hole punches, depending on the leaf. These kits usually involve many steps. The ones that I've worked with are around 15 steps, including membrane and cell wall lysis, many pipetting steps, addition of solutions to trap the inhibitors, many cleaning and washing steps. And the problem is that in each of these steps, we lose a little bit of DNA. But in wood, we don't have a great amount of DNA, so we can't afford to lose it. Kemp et al. Reported losses of 21 to 50% of DNA along the process using DNA extraction kits. And they concluded that, I will read now, understanding how and where to reduce loss of molecules during extraction and purification will serve to generate clear and more accurate data, which will enhance the utility of ancient and low copy number DNA, which is our case, as a tool for closing forensic cases, for example. So reducing the DNA loss is in general a big deal, even for leaves where almost all the cells have DNA. But as I said, we work with wood and we can't afford to lose DNA. So the fewer steps, the better for us. So I will advance to our question, that is, can we isolate only the living cells and avoid the dead materials that hinder DNA yield? Well, we tried to answer this question using wood anatomy. We know that the DNA is in the parenchyma cells that I said before, they were living cells. And we know where to find the cells. So instead of separating the cells from the vascular tissue, we applied a procedure that we called microcapture, in which we mechanically isolate each nucleus separately. This helps avoiding the inhibitors, but then it creates another problem, because I told you that the amount of DNA is not enough to go through all that extraction and purification steps using DNA kits, because we lose DNA during the process. And now I tell you that we used a single nucleus. How is this? Is even less DNA. How would this work? Because we are not adopting the DNA extraction kit. So we are not going through all that steps to extract DNA. We applied an extraction protocol that was developed to extract DNA from single cells. And then we can skip all the steps required when we use DNA extraction kits. So in this extraction protocol, we use a buffer to break the nuclear envelope inside the same tube in which we perform a PCR, which is the step where we copy the DNA. I'll explain this later as well. So we don't have to change tubes. We don't have to pipette and go all over that steps. So although we have less DNA, we don't lose it. This idea of extracting a single nucleus came from the assumption that if we have an intact nucleus, the DNA will be undamaged. And if we apply a process that requires minimal manipulation of this nucleus and steps, we'll be able to maintain the initial DNA quality. So what is DNA quality? Let me try to explain this inspired by the way Alex describes it. I'll try not to use technical words. So information in DNA is linear, the same way as we write sentences. Imagine that the information on how to make a tree, for example, and maintain this tree is a recipe written in a recipe book. So we have the cover of the book that is protecting the pages where the recipe is written. Let's say that the book is the nucleus, the cover is the nuclear envelope, and the pages are the DNA. If you don't take care of this book, with time, this book will get old, the print might fade a little bit, and if it's in contact with water, it might rot. Someone might rip one of the pages away. And we will start losing steps on how to make the recipe. So that's what I mean when we talk about DNA degradation. This linear message starts to break apart, and you get to a point where you have all of the words shuffled and it doesn't make any sense anymore. So DNA quality is about not breaking up the paragraphs into little pieces. Another analogy about this figure is you don't need to read all of the book to know what tree it is talking about. There will be one page where the name of the tree is written, and to identify wood, we just want to have at least that page intact. I'll talk about this a little bit more. Another concept that I want to show is that is the PCR that I mentioned before as a step in which we make copies of DNA. So recently everybody was talking about the PCR COVID test, and it's like photocopying DNA. For us, it is important to photocopy just the page where the information that says the name of the species is written, because we want to identify it. So we use little chemicals that start at the beginning of the paragraph and at the end of the paragraph, and will let us copy only that page. So every time we make a copy of the page, the copy can be used as a template and can be copied again. And so we double our quantity every time. So after many cycles, the photocopying we will have at the end basically only that page that we are interested in. And again, if this paragraph or if something is missing in this page, we will not be able to photocopy the whole page. So we will lose information. That is what we talk when we talk about DNA quality. So now I present to you the microcapture and single cell protocol. This method will help us overcome two of the limitations of wood as a substrate. First, the low quantity of living cells, focusing only on that cells that have DNA available for extraction. And second, the presence of the PCR inhibitors, targeting only the nucleus and avoiding contamination by extractives. Here is an overview of the protocol from preparation to species confirmation. But I want to let you know where we are going. So I will start by making the slides of the wood, finding the nucleus, grabbing it, squirting it into a PCR tube, photocopying it, taking that photocopied DNA and reading what words are in that paragraphs, and then taking that paragraph and pasting it to a gene bank that you can make an analogy to Google. When you copy a paragraph and paste to Google and you want to find the text, so we paste to gene bank and then we find out what species the book is about. Now I want to tell you a little bit about the species that we used. We used Carya, Ovata, Antilia americana. Carya is a medium to high density ring porous hardwood, and Antilia is a low density diffuse porous hardwood. And we used fresh wood, fresh leaves, and old samples stored in the Forest Products Laboratory in the MedW wood collection. And to show the applicability of the method, we also chose a softwood, and in this case was spruce, collected in 2015 and stored under freezing conditions. So let's go to visualization. We sectioned 15 micrometers of radial sections of each of the species and treated with DAPI. DAPI is a stain used for fluorescent microscopy that binds to double-stranded DNA and fluoresces. Fluorescent microscopy is basically a fancy and precise black light that, remember, that black light that makes our teeth and white shirt glow. Here we can see some differences in the species. So in Carya, the cells are taller but shorter in length, while in Tilia, they are shorter in height but longer in length. Another difference is about cell contents. In Carya, we can't see the contents under fluorescent light. But in Tilia, the contents are also fluorescent. And many fluorescent components, compounds, have cyclic compounds in their structure, like phenolic compounds that inhibit the PCR, which is the step that we make photocopies of the DNA. And here is Picea. In Picea, we have this big and long nucleus. It's taking almost all the height of the cell. Now, let's see the microcapture technique. This is the basic setting for the microcapture. This is an inverted microscope. The section lies on the top of a thin glass slide, and we use a micropipette attached to a micro injector and control it with a micro manipulator. This micro injector turns the micropipette into a tiny vacuum cleaner. This will suck up the nucleus, as we will see in the next slides. So this is how it works. We have a radial section. I always work on the very border of the section. I start by using fluorescent light to find the nucleus. So the nucleus is this round, glowy thing here. Then I switch back to light microscopy to position the tip of the micropipette in front of the open cell where the nucleus is. Then I lower the pressure inside the pipette using the controls in the micro injector, and I capture the nucleus. Finally, I switch back to fluorescent light to confirm that the nucleus is inside the pipette. Here we have a video. Here, this is Celtis. There is the open cell and the pipette. This tiny thing here is the nucleus, and we will see it being sucked up into the pipette. There you go. And now I'm getting the pipette out of the cell. And I'll turn the light microscopy on again. And there it is. So this is a small video. This is another video. This is Spicia. And here we did a little different because the nucleus was almost the same size of the cell height, as I said before. So we used the finest pipette. And because the nucleus was sticky, it stayed attached to the external wall of the pipette. Like that. It's cool, isn't it? So let's recap. Considering micromanipulation and microcapture, we included three different species of different groups. So there were 2 hardwoods, 1 softwood with different densities. We had fresh and old samples with different amount of parenchyma cells. So we expect that this method will be effective for other wood species as long as they have an intact nucleus. So the next step is the transfer. So with the nucleus inside the pipette, I remove the holder very carefully and then I insert the pipette into the PCR tube to release the nucleus. With the nucleus inside the tube, I will add the chemicals so that the machine can make hundreds of copies of that page that I want. That's the PCR that I talked about before. And at the end of the process, if I look at the tube, nothing changed visually. And in order to see if the copies were made, I add a little bit of this PCR product to a gel to confirm that there is DNA inside the tube. I'm not going to explain this process now, but I can come back later if anyone has any question after the presentation. But this is just to visualize that we had copies of DNA in the tube. So after we had a visual confirmation that copies were made, we had to make sure that it was not a contamination. So we sent our samples to the University of Wisconsin Biotechnology Center for the sequencing, which is that step where a machine will read the copies that we made before and tell us what it was written in the page. Finally, in the last step, it's where I paste that text into GenBank to find a match. So pretend GenBank is Google, but GenBank is actually a database with billions of DNA sequences. Here is the text, which would be, in our analogy, the story written in that page that we copied many times. I paste it here and do what we call a blast search. And this is the result. In this case, the sequence was Carry-Alvata, which is what we expected. And I want to address here that the goal of our work was not to identify this wood species. The reason we did these steps was to make sure that this DNA that we saw here in the gel was not the result of a contamination. There are some observations about this method. To be able to copy that region in the DNA, that we needed to have an intact nucleus. Here is an example. We have Picea in both images. On the top, we can see that the nucleus is well delimited. It has an envelope and the envelope is intact. Whereas here, this another Picea sample, it seems that the nuclear envelope is disrupted and the DNA is dispersed inside the cell. So what happened is that we failed to copy DNA from this disrupted material. First, we couldn't get all of this substance, all of this DNA out of the cell. And second, probably this was chopped in little pieces and we didn't have that whole page anymore that we needed to tell what species was that. So in our work, we observed that the microscopic anatomy of the nucleus was an important predictor of successful DNA amplification. Based on this, we assume that inside an intact nucleus, we have the best quality DNA that we can achieve. And if we transfer this intact nucleus into that PCR tube, we are not causing any damage to the DNA because it's protected by the nuclear envelope. We don't perform any mechanical action, and then we assume that the DNA prior to PCR is the original DNA quality, and that is the best quality that we can extract. Now, I'm going to briefly mention some applications of this method. And I want to add a disclaimer here about the images that I'm going to show. They are only examples. We didn't use this method to extract DNA from these materials that I'm going to show in the slides in the section, okay? So wood itself is not the most compliant material to extract DNA, as we've seen, especially if they have lots of extractives that inhibit the PCR. So this method could be useful when extracting DNA from recalcitrant wood, meaning that the species is difficult. It doesn't help us in the process. There are situations in which we don't have enough material to keep trying an extraction indefinitely. At the same time, most of those DNA kits call for hundreds of cells, if not more, as a starting material. We saw that they ask for 50 to 100 milligrams of material. So when we are working with this trace evidence, we don't have enough material. And using one nucleus at a time could help in the identification of trace evidence from a crime or for an accident, where we could get DNA from a slice as tiny as these ones here. Or from cultural property, like museum objects, in which we can't cause a notable intervention in the art piece. We also have several wood collections in the world holding material from trees collected hundreds of years ago, from populations that might be already extinct. And we could have genetic information from these ancient populations that don't exist anymore. The same can be said from herbarium material. both herbarium and wood in a collection are very precious material, and a method that enables us to use just a tiny piece is very desirable. So in summary, we saw that wood is a poor source of DNA. Also, there might be extractive that prevents successful PCR. So we suggested a method to capture a single nucleus that worked for different types of wood. But we also warned that there must be an intact nucleus for this method to work, and it is an application. This application is promising when we have small samples that we can't damage. I would like to thank everyone that joined us today, and also my co-authors in this project. If you have any questions, I'll be happy to answer now. Thank you.
Adriana, we've already got a couple submitted in the chat box, and then we'd certainly invite anyone that would like to unmute and ask a question. They can do that too. So let me pose the first one of these to you. It was from Julie. It says, amazing. One question, old, but were they frozen or were they on the wood collection? And I think she was talking about the extraction.
Yes, these were in the wood collection with no preservation method, anything, just old wood in the collection. And we were able to extract DNA from samples that were there all collected over 100 years ago.
And I was going to ask, are there limitations either in age or in the condition of the sample that affect your ability to be able to test?
Yes, so as I said before, we have to have a nucleus, an intact nucleus to perform this technique, but we can make a section and treat with DAPI, that substance that I mentioned before, and we look at the microscope. So if we have nucleus, then we'll be able to extract a nucleus and we can extract DNA from it.
Okay. And the next question came in from Brunella. It said, congratulations, it was a great presentation. I learned a lot. Question, we know that anatomical features vary from each species. How does the size of, and I'm going to mispronounce it, how does the size of parenchyma cell could affect this methodology? Should the method be adapted?
Okay, good question. So I might have a slide here to show you. I didn't put on the presentation because it would be too much to talk about, but we had these pipettes with different sizes, and this is the original pipette that we can break in different areas of the pipette, and we can make different tip sizes. So for each species, we need to know the height of the raised cells, because the pipette has to have an outer diameter small enough to be able to get inside the cell, but the inner diameter has to be big enough so that the nucleus can go inside the pipette. So that's something that for each species, we need to know the size of the nucleus, the size of the cell, so we can have pipettes to use for these different types. But as I mentioned before, for Picea, which is this image here, because the nucleus was at the same height of the cell, we couldn't make a pipette that worked. So we used the original one, and I showed in the video before that this nucleus had a substance, a sticky substance that got stuck in the top of the pipette. That's it.
All right. Any other questions? Here's one. This one comes from Hedda. It says, very interesting. You might have mentioned it, but when using old dry wood, does the wood need to be soaked in a liquid to be able to extract the DNA?
No. Well, we make a section, and then in the slide, I add just a drop of DNA-free water, but just enough to make the section wet. It's not totally dry, but I don't put anything or any other liquid, but it has just a little cover with DNA-free water.
Richard says, amazing presentation. I'm just glad that I was able to have seen this being done live. So, and I think the videos were really cool. So that was neat. Well, if we don't have any additional questions, on behalf of Forest Products Society, we'd like to thank you all for joining us. We'd especially like to thank Adriana for presenting. Please stay tuned for follow-up emails from us about our next Forest Products Insight, and certainly check the FPS website. We've got 3 conferences coming up over the next few months. We would certainly welcome your attendance there. So with that, we will sign off. Thank you, everyone, and have a great afternoon.
Thank you.
Okay.
Good afternoon. Bye.